We're extremely fortunate. For the past 30 years, thousands of cutaneous melanoma patients and their families have provided samples, answered questionnaires about their phenotype and environmental risk factors, and allowed us to follow their clinical course of their cancers. These incredibly generous and cooperative melanoma patients and their families have contributed greatly to the understanding of cutaneous melanoma. In the past 6 years, in collaboration with the Queensland Ocular Oncology Service, we made a concerted effort to recruit ocular melanoma patients and their families, particularly focused on uveal melanoma. The bulk of the recruitment takes place through Bill Glasson's Sunil Warrior and now Lindsay McGrath's Ocular Oncology Clinic at Terrace Eye, where both new and previously diagnosed patients are invited to participate in our research. Where possible, we collect an array of biological samples, tumour tissue, blood and saliva. And if required, we also endeavour to obtain paraffin blocks. However, archive samples are difficult to obtain, often having been destroyed, and when available are frequently unusable due to their age and poor quality. Obviously, as everybody else has spoken about today, we endeavour to have a patient-centred approach, and all the other factors, of course, are very important to us. Overwhelmingly, our patients are extremely motivated to participate in research. Typically, in clinics, patients provide informed consent and a saliva sample along with their family history. Each patient's journey is different. I attend all oncology clinics and surgeries. So, as part of a team, I'm able to gauge the appropriate time to discuss participation in research and the possibility of biobanking their tumour tissue, if that's what's required, or just simply talk about their diagnosis. As part of our recruitment process, participants are provided with a study questionnaire sheet which outlines their phenotype and environmental risk factors, as well as a questionnaire on their family cancer history, which also provides details of their relatives' contact details. Where possible, germline, blood or saliva samples are obtained, and tumour tissue, which is most commonly derived from enucleation specimens. We write to each relative and with the same questionnaires about skin colour, hair colour, eye colour, other risk factors, and if a relative is thought to be affected by a melanoma or any other type of cancer, we also attempt to obtain a germline sample. The Oncogenomics Laboratory has 2 databases. The commercial software Progeny manages clinical, family history, phenotype, environment factors, as well as pedigree data. This is where we track specimens, link all documents, including consent forms, pathology reports, and all correspondence and clinical information. As well, we have an Access database created to record all cancer incidences using the International Classification of Disease for Oncology codes, along with histopathological features, and many of them in line with what Svetlana was discussing. Here we also collect or record causes of death using the International Cause of Death codes, reflecting both cancer and non-cancer causes of death. Our primary goal of verification is of all cancer incidences within each family. Once finalised, cancer confirmation is entered into the Access database, noting the degree of confirmation, such as whether it was a histopathology report or a death report or personal or family recollection. Verification is achieved by regularly visiting the Queensland Cancer Registry and obtaining cause and cancer and cause of death information, and we also liaise with other Australian cancer and New Zealand cancer registries. We chase pathology reports, obtain clinical confirmation from clinical records or death certificates. Sometimes this can be a very slow process, though overall we acquire good quality, reliable data. Within our team, it works well that we have people with specific roles, and we always ensure that all data is cross-checked to minimise data entry errors. We discuss discrepancies with what participants say and what we find, and with agreement produce the cleanest possible data. So this is an example of a pedigree that's gone through the whole process. So the proband would have been Elizabeth, who has a confirmed melanoma at the age of 40, diagnosed at the age of 40. She's donated blood and tissue samples, and of course the C indicates that she has a consent. So as soon as we look at a pedigree, we can see what's happening with that family. She informed us about her siblings and her her children. And so we wrote to Brother John, and John gave us a saliva sample, and we confirmed that he had 4 cutaneous melanomas. We also confirmed that his daughter Kate, who also provided a saliva sample, had a breast cancer diagnosed at 35, and Jack told us that he didn't have any problems, but he did provide his consent. Proxy consent was obtained for Dad Joseph, and we found that Joseph had a melanoma, cutaneous melanoma at age 50, which is why he's coloured in black, and he died at the age of 70 and had a prostate tumour diagnosed at 68. Now, whether he died of his cutaneous melanoma or his prostate cancer is all recorded in the database. And obviously we have a lot of contact with these families to get this, and so it's these one-on-one trusting relationships that extend through our whole cohort and contribute to the ongoing enthusiastic participation and support of our research. This is just a quick back-of-the-envelope sketch of the variety of general cancer types of over 500 patients and on our books and their family members, though not all of them do we have germline or phenotype data on. What I present today is a work in progress of 431 patients whom to January this year provided germline DNA samples and answered questionnaires. 226 male, 52%, and 205, 48%. Female, age of onset was between 13 and 91 years. 271 of these patients, of whom, along with their families, had completed the described data collection and verification process. The cases reported type 3 Fitzpatrick skin, and you'll see approximately equal numbers had type 1 and type 2. Similarly, whilst the majority have blue eyes, the other substantial subset are neither blue nor brown-eyed. Of the 280 participants who provided information on keratinocyte skin cancers, that is basal and squamous cell carcinomas, 181, 64% in pink, were affected. With a range of associated cancers in this cohort, so far 59 people, so far 59 individuals have developed one or more cutaneous melanoma, and 25 of these people in green have both and cutaneous melanoma. With both and cutaneous melanoma also had another type of cancer, so they had 3 or more cancers. 2 participants were identified with first-degree relatives affected with All 4 individuals are included in this analysis. Another 2 participants included in this analysis had second-degree relatives with These families are being assessed for the presence of BAP1 germline mutations or other potentially novel high-penetrance susceptibility mutations. These interim analyses of the Queensland cohort reveal a noteworthy proportion of patients with other cancers. Excluding keratinocyte cancers, 123, around 30% of this group have and at least one additional primary cancer. The association of cutaneous melanoma, and a variety of other cancer types suggests a generalised predisposition to cancer in the Queensland population. As described, this is an interim analysis, and we understand the limitations and ascertainment bias, and we look forward to expanding and continued surveillance of our generous participants. Replication of this data and analysis in other populations would be of considerable benefit. The Queensland Ocular Oncology Service Access Database at Terrace Eye maintains a chronological record of each patient from initial presentation, including clinical features, tumour location, visual symptoms, key diagnostic factors, treatment details, response to treatment, adverse events, and along with the incidence of progression to metastases or other cancer types are also recorded when available. Our work is always patient-centred. Our participants are the strength behind the science. Our foremost aim is to make the process as simple and as unobtrusive as possible. We always leave communication open with our participants, allowing for future follow-up, and the provision of accurate, clean, detailed data that can confidently be used in tandem with laboratory research results is our bottom line. We are obviously indebted to all our participants and collaborating clinicians and the many allied health professionals without whom we would not have been able to conduct our research. Thank you.